Okay, sir.
Good evening, dear doctors. This is our second discussion on the MCQs based on the biochemistry, the subject biochemistry. Today, we will be discussing about some questions from protein metabolism, mineral metabolism, and nuclear protein. In other words, we will be discussing about nucleic acid metabolism. Okay, anyway, I would like to tell you that the hard work, which is prolonged hard work, that means perseverance, is very important for any success. So I hope you were studying, you were revising the topics day by day as per our schedule, as per our timetable, what has already been given to you. So this might be going to help you out from your discussion part, maybe as a revision, it might be useful. So straight away going to my topic today. First question is on protein metabolism. First question is, which of the following is a derived protein? So this question is about classification of proteins. So as you know, the proteins are classified into simple protein, conjugated protein, and derived protein. We'll try to say some examples and some importance for it. In this one, anyway, it's a very simple question. Protamine and prolamine are, and even lactalbumin are simple protein. The albumin, what is present in the milk, we call it as lactalbumin. Protamine and prolamine are another types of simple proteins. And peptone is that means a protein on hydrolysis. Any compound produced part of the hydrolysis of a protein, we call it as derived protein. So here, peptones are produced by during the hydrolysis of protein, and so peptone is a derived protein. Now we'll just have a look on the classification part. As I said, simple protein, conjugate protein, and derived protein. Derived protein, we have peptides or peptones. Based on the number of amino acids, we just call it as peptides and peptones. Okay, when we think about a simple protein, in that, first one is albumin. It is soluble in water and coagulated on heating. And second one is globulin. It is insoluble in water, but soluble in dilute salt solution. They are globular protein. What all things what I'm going to say is globular protein only. And third is protamine. Here, I would like to say an importance of protamine. Protamines, they are soluble in water, dilute as an alkali. They are arginine and lysine rich. See the importance of that. The protamine zinc insulinate is the one what we get in the market, in the commercially. One of the way by which insulin is provided in the medical stores, in the market, in the commercially available is as protamine zinc insulinate as a complex. And also another use of protamine is that it neutralizes the anticoagulant effect of heparin. So whenever heparin overdose is there, you will be giving protamine for that. Then another type of simple protein is prolamine. They are soluble in 70 to 80% alcohol. They are rich in proline. And the gene of corn, gliadin, what is present in the wheat, is prolamine percent. And some persons have an allergic to this prolamines. That's why they have irritable bowel syndromes and celiac diseases because of the prolamines. And another one is lectin. What have a high affinity for sugar, carbohydrate. This is used in cell signaling pathways and all, and is soluble in 30 to 60% ammonium sulfate. So this is about some different types of simple protein. And then one more is there, scleroprotein. It's a, what do you call it, globular protein, and other one is soluble in strong acid. Okay. Then next question. So we said about some simple protein. Conjugate protein, we know nuclear protein, lipoprotein, phosphoprotein. These all comes. Whenever a non-protein part is attached to it, that's called as conjugate protein. So simple protein, conjugate protein, and derived proteins. Now, and next question. Tertiary structure of protein is maintained by all except. So this question is on structure of protein. They are trying to measure your idea on the structure of protein. As you know, protein have a complex structure So we always classify or we just divide the structure or study the structure at 4 levels. One we call it as primary, second secondary, third tertiary, and quaternary structure. What do you mean by a primary structure? Then if a protein is study, the simplest thing what you're supposed to say is how many amino acids are there in a protein and in which order they are arranged, they are linked through peptide bond. So primary structure defines the number and sequence of amino acids. Okay, so that's all about the primary protein. And their primary protein is maintained by covalent bond, mainly peptide bond. Of course, disulfide is also a covalent bond, and that is also there in that. So primary structure maintained by peptide bond and disulfide bond. And secondary, tertiary, and quaternary structures are maintained by non-covalent bonds. Along with that, that's the point. Along with that, so they are maintained by non-covalent bonds like hydrogen bonds, hydrophobic interactions, ionic bond, disulfide bond, and van der Waals force. These also there. And along with that, covalent bond, disulfide bond is also there. So that's a tricky part of this question. Secondary, tertiary, and quaternary structures are, we always say in general, they are maintained by non-covalent bonds, but along with that, disulfide is also important. Okay, so in this condition, all the 4 are there to maintain the tertiary structure, and so it's none of the above is the answer. Now we're coming back to the point. What do you mean by a secondary structure? Secondary structure means, see, in a protein, in one part of a protein, polypeptide chain, how it looks like. That's called the secondary. Okay, local configuration of a protein, local configuration, locally, one part how it looks like. And we have 2 types of secondary structure. They are alpha helical structure and beta pleated structures, beta pleats. So these are 2 major secondary structure. And then what about the tertiary structure? Tertiary structure means, of course, by alpha helical structure, there's a coiling. Again, a super coiling comes. So totally, tertiary structure means a 3-dimensional structure of a whole protein or a polypeptide chain. They call it as tertiary structure. And then what about quaternary? Again, I'll say primary means the number and the sequence of amino acid. Secondary means and they are maintained by covalent bonds. Secondary means local conformation in one portion, how it looks like. It can be a helical structure or can be a zigzag structure like that. Tertiary means not locally, totally all together, 3-dimensional structure of polypeptide chain. And what about the quaternary? In some proteins are made up of more than one polypeptide chain. So, for example, hemoglobin, we have 2 alpha chain and 2 beta chain. Immunoglobulin, 2 heavy chain and 2 light chain. After synthesis, they all will come together, aggregate together. So, what are the interactions between them? So, when there are more than one polypeptide chain, then that protein always assumes one more level of structure called as quaternary. From the picture, I'll try to show. So, if this is a protein, hemoglobin, 4 chains are there. How they are linked together, they are close together, how many hydrogen bonds are there, and what are the interactions between them. That are explained by the quaternary structure. So only protein having more than one peptide chain will be showing a quaternary structure. Now tertiary means one peptide chain. How it look like, how totally 3-dimensional structure of that. That's called tertiary. Then one part of that. One locally, how it look like. So here we are just showing about the alpha helical structure. And then in that, how the amino acids are arranged. In what are the amino acids present, in which order they are present. That's called as a primary. So that's what. So primary is maintained mainly by covalent bonds, and secondary, tertiary, quaternary are maintained by non-covalent bonds. It can be hydrogen bonds, hydrophobic interactions, ionic bonds, van der Waals force. Along with that, the tricky part, it is also maintained by disulfide bond. Okay, and one more point. During denaturation process, only the primary structure will not get altered. All other structure gets altered. Okay. Next question. Indole ring is present in Now the question is about amino acids, about the structure of amino acids. Indole ring is present in what? That's a question. So it's present in the chapter. We'll just come across now indole ring. So you are supposed to study the structure. So we'll try just go for its types of classifications of it. So classification of amino acids. How many amino acids are there? We have studied just 20, but now two more has been identified. I'll come to that word. Okay. So first is about on the structural basis, we have aliphatic, aromatic, basic, acidic, and one more. Not amino acid, we have amino acid. Proline is an amino acid. That's one important question you should not. Amino acid, one of the fact, amino acid proline. So that's one classification. I'll come to that terms later. Next, second, in the aliphatic itself, we have a specific type called as branch chain fatty acid. We have amino acid containing hydroxyl group, amino acid with sulfur atoms. It goes like that, containing two acid group, glutamic acid and aspartic acid, containing amide group, glutamine and aspartame, containing basic group, two amino groups, lysine and arginine. So it goes like that. Another classification is essential, non-essential. As you know, the definition that cannot be seen inside the body but should be supplied through the diet, we call it as essential. And that can be synthesized in the body, we call it as non-essential. We have 8 essentials and 2 non-essentials. Study the names, 2 semi-essentials. Then another classification is about glucogenic and ketogenic. In that, one question there, purely ketogenic. That means from which glucose cannot be synthesized. Is purely ketogenic, is leucine. Okay, so I'll just go through that now. In that, our classification about the structure, we got to have the aliphatic, simple amino acid. The simplest is glycine, okay? Simplest is glycine, and the alanine is also a simple amino acid. Branch chain, valine, isoleucine, and leucine. We need it later also in another type of disorder. So study it as VIL, valine, IL, VIL, valine, isoleucine, leucine. Then hydroxyl group, OH group containing amino acids, serine and threonine. Sulfur containing cysteine, and 2 molecules of cysteine combined together and form cysteine, and then methionine. Then with amide group, asparagine, glutamine, and dicarboxylic acid, that means acidic, aspartic acid and glutamic acid, and diamino, that means 2 amino groups minimum, lysine and arginine. And this one is also basic. So name basic amino acid, lysine and arginine. Of course, one more is that histidine also. Okay, that's one classification, type of classifications. Then another next one is about the aromatic. Aliphatic is over, aromatic. In that phenylalanine, tyrosine, tryptophan are aromatic. And as I told, there are some containing more than one cycle structure and cycle structure with other than carbon atom. They are called as heterocyclic. So we have tryptophan and histidine. As I said earlier, we have an amino acid proline. And there are some derived amino acid, what are present in the protein. Our hydroxyproline, hydroxylysine, gamma carboxylated glutamic acid, acetylated or methylated histones, amino acids in body present in the histones. There are some derived amino acids. And we have some derived amino acids what are not present in the protein, ornithine, citrulline, homocysteine. And there are some non-alpha. We all, so far I said is alpha amino acids. So 20 amino acids are alpha amino acids. Okay, that means amino group at the alpha positions. We have some amino groups, we have some compounds having amino group other than the alpha positions. Gamma amino butyric acid, GABA, beta alanine. These are some examples for non-alpha amino acids. Now the next question about who all are the glucogenic, ketogenic, and both glucogenic and ketogenic. Leucine and lysine are ketogenic. In that, lysine, on its breakdown, will combine one more compound and will produce the 2. One will be a keto group, another one that can be goes for glucose. So, lysine also comes under ketogenic. But when somebody asks you what is purely ketogenic, it is leucine. And then tyrosine, tryptophan, phenylalanine, isoleucine can be called. All the 4 are both glucogenic and ketogenic. and other amino acids are totally glucogenic. Okay, one thing. Now another one is about, as I said about essential essentials earlier. Then you should know about this one. After identify the 20, we have 21st 2 and 22nd. 21st is selenocysteine, selenium containing. Instead of sulfur atom in the cysteine, selenium is attached. So selenocysteine. And 22nd, another one, some modified form of lysine, pyrrolysine. That's another one mark question can be about the amino acid classification. As I said earlier, purely ketogenic is lucid. Derivatives we have already said. What are derived forms? And one more question about the amino acid. Amino acid with maximum buffering capacity. Is there an acid balance? Histidine has the maximum buffering capacity because it's side chain, imidazole group, have a pK value near to a body pH. So histidine is having maximum. So that's also another question for you, another factual question. Amino acid with maximum buffering capacity. Okay. Now about the compounds produced from each compound, each amino acid. First one, from glycine, what are the compounds produced? Out of all the things, hemoglobin, in the sense you should understand heme, glutathione, purine, creatine, and creatinine. See, all these compounds for the synthesis, we need glycine. For purine, should know always, that is actually a question there. We have a structure of purine that C4, we call it a C5 and N7. The whole glycine is attached. Carbon atom at the 4th position, carbon atom at the 5th position, and nitrogen atom at the 7th position. These all are provided by the glycine. Should know that. C4, C5, and N7 of purine. Then glutathione is a tripeptide. Gamma glutamyl cysteine glycine, called as LAG. Nowadays, all of us are very familiar with this word. It's an antioxidant. Glutathione is an antioxidant. We call it as gamma glutamyl cysteine glycine. So at one end, we have a glycine. Gamma glutamic acid is here. So carboxyl and glycine. In between, cysteine comes. So 3 amino acids. It's a tripeptide. Tripeptide antioxidant. Okay, then creatinine. What's the use of creatine? In our body, in the muscle, creatine is stored as creatine phosphate. That helps in the synthesis of ATP. That reaction we call it as Lohmann's reaction. So our creatine is stored in the body as creatine phosphate. That helps in the synthesis of ATP in the muscles through a reaction called as Lohmann's reaction. So all the 4 major compounds are produced from glycine. Any other functions of glycine? The last discussion we said about that, about the detoxification process. Some compound, as I said earlier, in the jam and fruit juices, we have a preservative benzoic acid that can be excreted only by conjugate with glycine to form hippuric acid. So another function of glycine is it helps in the detoxification. Not only benzoic acid, one more be a bile acid, folic acid. For the conjugation purpose, we use glycine. And one more function, it's a neurotransmitter. So glycine, so here, all the options above are right. And other functions, glycine helps in for conjugation and also as a neurotransmitter. Okay. Next one. So now we'll just go for the other amino acids. Major compound but from other amino acids. You should know about that also. From the phenylalanine and tyrosine. Phenylalanine, one step converts to tyrosine. Okay. Phenylalanine is essential, tyrosine is not essential because if we have phenylalanine, tyrosine can be synthesized. From them, our color pigment, our melanin is produced. Tyrosine, rhymically, thyroid hormone is produced. Then catecholamine, dopamine, epinephrine, norepinephrine. These are the compounds produced from phenylalanine and tyrosine. And one more vasoconstrictor, we have tyramine. What about tryptophan? At least you might be remembering from the last class, we were discussing about that. From 60 milligram tryptophan, we can synthesize 1 milligram of niacin. Yeah. So one vitamin niacin and neurotransmitter serotonin, melatonin. These are three major compounds. Of course, one carbon. We get a one carbon group from it. A one carbon group. And using that, we can synthesize. We can go for DNA RNA synthesis. One carbon compound is another thing about tryptophan. Okay. Only these three things you should know about the major biologically important compounds produced from glycine, phenylalanine tyrosine, tryptophan. From methionine, we get cysteine. From cysteine, we get taurine, another compound that helps in the conjugation of bile acid and bile salt. And then about glutathione also contains cysteine. No? So from cysteine, we can synthesize taurine. For the glutathione we use. That's also you should remember. Okay. Next question. What are the sources of ammonia in urea cycle or urea? So the question is about urea. About urea synthesis, urea cycle, what is its importance? And one more question I put here. How many moles of ATP is produced when during the synthesis of this one, urea? Okay. What is it about? What the first should know about urea cycle? Urea cycle. Urea cycle, what is the use of that? It is to detoxify ammonia. During amino acid metabolism, amino acid breakdown, the amino group will be removed as ammonia. Okay, from an amino acid. So understand that way. Amino acid on its breakdown, it produces ammonia. This ammonia is highly neurotoxic. So somehow it should be detoxified. So first in the cells, what happens? In the brain, what happens is suddenly this ammonia will combine with glutamic acid, just GLU stands for glutamic acid, and form glutamine, amide form, glutamine. And that will be transported towards the liver. And from other tissues and mainly in the muscle, what happens is from the muscle, this ammonia, again neurotoxic. So what happened? Ammonia will combine with pyruvate, and will convert to a compound called as alanine. Now what happened? These glutamine and alanine will be transferred towards liver. So my first point is that the ammonia that is produced during amino acid metabolism is neurotoxic. So the first line of defense is that it converts to glutamine in the brain and convert to alanine in the muscles. Then it's transferred to liver. In the liver, this ammonia is converted to urea. This ammonia will be converted to urea. Okay. So urea cycle, the first point, urea cycle takes place in the liver, first point. Second is that it helps, the urea cycle is to detoxify ammonia. Okay. What happens as soon as this glutamine and alanine comes in the liver, it converts to ammonia and glutamic acid. They go back. That means with the help of glutamine is enzyme, convert back to glutamic acid and ammonia. Okay. So one of the source of ammonia, so urea cycle for the ammonia is just ammonia itself. Okay. Another source is what? I'll just show that this one. So anyway, I'll go to the urea cycle. You'll understand that. Here, just let me go for the answer. Okay, fine. This is what urea cycle take place in liver. Uses to detoxify ammonia. First one is about the source of ammonia. One is, I'll show the cycle later. Source of ammonia. One is ammonia itself, that is produced from either from glutamine or alanine. Another is our aspartate. I'll show that. So these are aspartate and ammonia. Ammonia are the source of nitrogen atom and the urea. So what is urea? I hope you understood that. NH2, sorry. NH2, C double bond O, NH2. One NH2 from ammonia, one NH2 from aspartate. And then how it transported? It's transported towards liver as glutamine or as alanine. And energetics, I'll just show the picture, then you understand that. Whenever you ask about energetics, thereby we are asking, does it have any relation with the TCS cycle? I'll just show that. And what's the clinical importance of urea? This is what you're supposed to know. Now listen to this one. Our ammonia plus carbon dioxide, I'm sorry, I added there, plus carbon dioxide giving carboxyl phosphate. So there's first amino group. And second ammonia is from the amino group is from aspartate. And this aspartate, after it's converted to organosuccinate, one fumarate is formed. This fumarate goes to TCS cycle. So in fumarate, in TCS cycle, it produces 2.5 ATP. During urea, now one question. Urea cycle, here 2 ATPs, here 2 ATPs are utilized. Here straight away 2 ATP is converted to ATP. And here 1 ATP is converted to AMP plus PPI. So that's equivalent to utilization of 2 ATP. This itself. So totally 4 ATPs are utilized. But when the fumarate, a product from the urea cycle, goes to the TCS cycle, it produces 2.5 ATP. Okay. So here, this fumarate gives plus 2.5 ATP. It gives, provides. But urea cycle as such, utilize 4 minus 4. So now the question, when one urea is produced, how many ATPs are utilized? It is 1.5. 1.5 ATPs because of its relation with TCS. Okay. And what's the use of what is the clinical importance of urea? Urea value, we always say around 20 to 40. 20 to 40 milligram per deciliter is the value of urea. What is the importance of urea? Both urea, blood urea, and creatinine are an indicator of kidney function. So that is an important one. As a part of renal function, you should know it. Kidney function indicator. So whenever there's a kidney dysfunction, both urea and creatinine value will elevate. Okay, so that's the clinical importance of urea. So we set up some points about urea cycle now, about what is urea, what are the sources of ammonia, what are the energy, how much ATP is utilized, what's the, how the ammonia is transported, what's the first line of defense against ammonia detoxification. This glutamine, conversion of ammonia plus glutamic acid to form glutamine is the first step of defense mechanism. Okay, that's about the urea cycle now. Now going for the next. In the physiological system, nitric oxide is known to act through. And nitric oxide is said to be in the last decade. It was a decade of the molecule of the decade, okay? Nitric oxide. From which compound is produced? You should know that nitric oxide is produced from arginine. Arginine with one enzyme, nitric oxide synthase, converts to citrulline and nitric oxide. Then one of the fate of nitric oxide is that it converts to high energy, highly reactive compound, peroxynitrate and nitrate. They are used by the macrophages to kill the microorganisms. Okay, so one of the use of nitric oxide, it helps macrophages, okay, in its action. What are the other functions? Okay, another function of nitric oxide is that through cyclic GMP, that's the second messenger for that. It helps in the relaxation of smooth muscle. Thereby, what's the use? One use, vasodilation. So that's why any person with angina pectoris, we give a sublingual drug in that can produce nitric oxide. So that will help, that will be able to reduce so that will be able to dilate our vessels. So pressure can come down, a relief for angina pectoris. Another function, it can prevent platelet adhesions. So these two, because of these two things, our endothelial function, we always measure by finding out the level of nitric oxide. Okay? Endothelial function, endothelium that helps in the vasodilation and platelet adhesion inhibition. Fine. So these two functions through nitric oxide. Now understand this one. Nitric oxide synthase is the enzyme. Is an enzyme that helps in the synthesis. There are 3 nitric oxide synthase. They call it as enos, that means enos in the sense from the endothelial cells. Another one, inos, inducible, that's present in the macrophages. And third one, neuronal, NNOs. And I want to say about inos. That is the one that's present in the macrophages, they are inducible. So whenever there's infections, macrophages will be able to produce this one and help in the production of nitric oxide. But one problem with that. So in the case of septicemia, you might have heard this thing, septic shock. In the case of septicemia, there can be a vasodilatation and the patient won't be able to recover from the hypertension. We call it as refractory hypertension. That means the patient is not responding to the medicines for the hypertension. Why? Because of nitric oxide. So just understand what happened. This nitric oxide, what is produced through with the help of inos, through cyclic GMB, it will be relaxing, helping the relaxation of smooth muscle. So thereby vasodilatation. So that's important thing you should know. And of course, this property of nitric oxide is vasodilatation. That's what we use in Viagra for the penile erections. Vasodilatation is essential. So nitric oxide in that way. So the Viagra contain a compound that can produce nitric oxide, that can retain the level of nitric oxide. Actually, it will be having inhibitor that inhibit the cyclic GMP. We have an enzyme called as phosphodiesterase that break the cyclic GMP. We have also Viagra contains some compound that inhibits phosphodiesterase so that the level of cyclic GMP will be maintained. Thereby, the vasodilatation will be maintained. So penile erections. Okay. So this is what happens. So now my point here is nitric oxide. Its action through cyclic GMP, help with the smooth muscle relaxation, thereby vasodilatation. And interstitial motility is reduced, sphincter become more relaxed. That's another function of it. Then the macrophages through the peroxynitrate. That's another function of nitric oxide. And one more function, it acts as a membrane molecule. Neurotransmitter act as a membrane molecule. So yes, I'm trying to conclude about the nitric oxide. Produce some arginine using an enzyme nitric oxide synthase. We have 3 types of nitric oxide synthase: enos, inos, and NNOs. E for endothelial. So endothelial function. Endothelium is the innermost layer of our blood vessels. That function depends on how much nitric oxide can produce. That way helps with the vasodilatation and the smooth, this one, prevents platelet adhesions. And then inos, a clinical importance is that whenever we have a severe infection, macrophages will produce through the inos, it produces nitric oxide that causes vasodilatation. So that leads to septic shock, refractory hypertension. Please note that. Another function, another clinical importance, macrophages help in the conversion of nitric oxide to peroxynitrate that help in its action. That's about the nitric oxide. Okay. Now, some disorders. So just try to set about almost all the what are the compound produced from an amino acid. If we were trying to say that. And now comes to some disorders related to it. See here, a disorder related to amino acid carrier defect. Okay. So we are going to disorder now. In that, from the here what happens? This is what about cystinuria. What is cystinuria? Cystinuria is nothing but 2 cysteine molecule combined to form cysteine, and 2 cysteine combines together to form cysteine. That is there in the urine. And here, understand this way, we have a transporter for cysteine, ornithine, arginine, and lysine called, study in that way. A transporter that help in the absorption and reabsorption of 4: cysteine, ornithine, arginine, and lysine. And if there's a defect in that, what happened? This will be present in the urine. So cystinuria occurs when a defect in the transporter, a carrier transporter, that transports 4 types of compounds. This one, cysteine, ornithine is a derived amino acid, arginine, and lysine. Other we will come across now. Maple syrup urine disease, I'll just say about that. That's a disorder related to branch chain amino acid. Name branch chain amino acid. Just now I said the branch to how to remember the branch chain amino acid. To climb the branch, we should have a willpower. Just understand that we will. So now you will be able to say that. Valine, isoleucine, leucine. For any amino acid, the first step of metabolism is transamination. Okay, that's a common thing, not related to this one. Transamination is the first process in the breakdown of any amino acid. After the transamination, it will produce a keto group. Amino group is removed, then keto group will be there. So we get alpha keto acid. So branch chain amino acid convert to branch chain alpha keto acid. For its degradation, we need a dehydrogenase enzyme. We need an enzyme. We call it as branch chain alpha keto acid dehydrogenase. This enzyme defect leads to a condition called as maple syrup urine disease. So in the urine of the patient, we'll be having the keto acids of branch chain amino acid. Highly fatal, maybe in the first year itself, or within 1 or 2 years after the birth, the child will die off. So here, there will be ketosis, ketoacidosis will occur, vomiting, severe vomiting because of branch. So, and here the urine will be having a smell of burnt sugar. That's why the word maple syrup or maple syrup. Smell of burnt sugar. So it's related to branch chain amino acid. Branch chain amino acids are valine, isoleucine, leucine. And alkaptonuria. What's the many of the word alk? We know about albinism. Alk means white. It's just opposite is that alk. Alk means what then? Black. Black colored urine. It happens in which metabolism? Tyrosine metabolism. Tyrosine metabolism. A compound is produced called as homogenesic acid. Genesic acid. And this, for its breakdown, we need homogenesic acid oxidase. Homogenesic acid oxidase. The defect of this one, deficiency of this enzyme, what happened? This converts to some black colored compound we call it as alkapton bodies. Black colored alkapton bodies and it will be excreted of the urine. So on keeping urine, when we void out, it will be containing homogenesic acid that's not colored. But on keeping, that become convert to oxidized to form alkapton bodies and the urine become black in color. So some black colored bodies in urine, from that word alkaptonuria, because of the enzyme deficiency, homogenesic acid oxidase comes in what metabolism? Tyrosine metabolism. And for the phenylalanine, the one step of phenylalanine is addition of phenylalanine to, we'll add a hydroxyl group with the help of NADPH and other groups and oxygen atom and form tyrosine. That's the first step of metabolism of phenylalanine. Okay. And in this one, this step, addition of hydroxyl group, enzyme name phenylalanine hydroxylase. Enzyme phenylalanine hydroxylase. And this phenylalanine hydroxylase deficiency, what happened? This phenylalanine will be converted to then phenyl ketones, phenyl acetate, like that. They are called as phenyl ketones. So phenyl ketones in urine, phenylketonuria. So that happened because of the deficiency of enzyme phenylalanine hydroxylase that converts phenylalanine to tyrosine. So that's about some disorders here, as I showed. So here the answer is carrier. Though all other three are about some enzyme deficiency, only one is having a carrier transport protein deficiency, that is cystinuria. Okay, so we go for next question now. Just a study on all some disorders related to the amino acid metabolism disorders. In glycine metabolism, we have two major compounds in the urine and disorder: hyperoxaluria type 1, hyperoxaluria type 2. That means oxalic acid more and more the urine. That comes related to glycine metabolism. two amino acid, two enzyme deficiency, alanine glyoxylate aminotransferase, glyoxylate reductase. These enzyme deficiency can lead to type 1 and type 2. And one more is there, non-ketotic hyperglycemia. In the glycine, I can't write here, a lot of things are there. So glycine metabolism, there is one enzyme for the help for the breakdown of glycine. We call it as glycine cleavage system. And that's the same, just opposite can also work. We call it as glycine synthase system. If that enzyme is deficient, then that leads to a condition, non-ketotic. There is no ketosis, but glycinemia, hyperglycemia. Is also there in the glycine. So there are three disorders related to glycine: hyperoxaluria type 1, hyperoxaluria type 2, and non-ketotic hyperglycemia. Next one is about in methionine metabolism. One enzyme, cystathionine beta synthase deficiency, leads to a condition, homocystinuria. Cystinuria. Homocysteine, two molecules combined together and form homocystinuria. Is related to methionine metabolism. And this is the one where the patient will be having, you can understand the patient. Very long bones will be very thin and so long. Another one, scoliosis. Vertebra will be some side, scoliosis. And in dental ways, we can say high arched palate. In homocystinuria, you can see high arched palate and crowded teeth. There are two manifestations. Dental manifestation, what we see in homocystinuria, high arched palate and crowding of teeth. Okay. Then maple syrup urinase, as I already said, it's about branched amino acid. Name branched amino acid. To climb a branch, we should have the will. Valine, isoleucine, leucine. And then enzyme deficiency, branch chain alpha ketone acid dehydrogenase. Then we have, just I said about cystinuria, where cysteine, there's a transporter called, I said about the transporter. And one more, again, one more. Cystinuria means this one, cystine is excreted through urine. Another word, cystinosis. I hope you know the word, you make it familiar. Acidosis. When an acid accumulates, we use the word acidosis. When alkyl accumulates, we use the word alkalosis. When a fluoride accumulates in the body, we use the word fluorosis. Same way, cystine accumulates inside the cell. What happened? There is a transporter to transport it across the cell, maybe through the lysosomes, and then we call it as a transporter for that, and a protein we need for it. I have written here, cystinosin is needed. Any defect in that, what happened? Cystine will be accumulating in the cells, in the tissues. That we call it as cystinosis. Then phenylketonuria I said, regarding the phenylalanine metabolism, enzyme is phenylalanine hydroxylase. Alkaptonuria, black-colored urine, related to tyrosine metabolism, homogenesic acid oxidase. Another one, I think I have already said about this one, heart disease in the last class discussion. Tryptophan, its absorption and reabsorption, not only for tryptophan, or for all the neutral amino acid. We have a transporter. That defect in transporter leads to the excretion of tryptophan and neutral amino acid in the urine. It was first identified in a family whose name was Hartnupp's, apostrophe S, okay, Hartnupp's family. So this is called as Hartnup disease. Again, that's also part of a transporter, okay, of neutral amino acids, and called a Hartnup disease. There you have, as you know, tryptophan. From there, we synthesize the amino acid niacin. So a patient with Hartnup disease, niacin cannot be synthesized, so they'll be having pellagra. That you have to remember. Okay, then for urea cycle, for each amino acid deficiency, there is one disorder. So I'm not writing about that. Just go through it. So these are some of the major disorders related to amino acid metabolism. Okay. Next one about a major protein. The major protein in our body is collagen. Okay. And the question, collagen enriched in what? As we know, 1/3 of collagen is glycine. And we have, and few here and there, 20% we have proline and hydroxyproline. So the answer is proline and glycine. So please note, collagen, the most abundant protein in our body. In the plasma, the most abundant protein is albumin. Please note, in the plasma in the blood, the most abundant protein is albumin in the plasma. But in the body as such, throughout when we compare all the, when we check out all the extracellular matter and matrix and everything, collagen is the most important one. You should know about its structure. It has first the unit of collagen we call it as proper collagen. It has a triple helical structure. I'm sorry, it's again hidden there. Triple helical structure. Fine. Triple helical structure. We call it as proper collagen. And it has a quarter stragate. That means 1/4. It is moving from the next one. It is not standing exactly. When we keep the coin one by one, we keep just above as such, right? But here it cannot. It will be, its collagen will be kept in such a way that it will be removed 1/4. So quarter staggered arrangement. So triple helical, we call it as collagen structure of collagen. We always say in this way, triple helical, quarter staggered. Quarter staggered arrangement. That's what we always say about collagen structure. And some disorder related to collagen and something more I am supposed to say, collagen about its modification. First, during its synthesis, first these chains as inside a separate one. Each alpha chain we have separated one. Okay. Then some areas will be removed. Then they clump together. So removal. That's one modification after protein synthesis. Protein synthesis, another name, translation. So modification after translation, post-translation modification. So one of the post-translation modification, and just like to say here itself, a big chain was there. Again, it forms in this way, triple helical structure. And then what happened? Both sides, some will be removed. We call it as proteolytic cleavage. One example is this one. Second is our dicing enzyme, pepsin, produces pepsinogen. Remove one part, become pepsin. Trypsinogen to trypsin. So they are example for proteolytic cleavage. Second type of modification after synthesis is that they all aggregate together. They all aggregate together. So another example, aggregation of chains is another post-translational modification. It happens for immunoglobulin also. Happens in the hemoglobin also. We have hemoglobin chains as I say separately. Then they clump together for hemoglobin. Immunoglobulin, we have 2 alpha, 2 heavy chain, 2 light chain. They are synthesized separately. Then they combine together. So that's aggregation. Then to link between these two, there is some hydroxylations. Or we have oxidation purpose. So covalently we are attaching something to it. So covalent modification, another modification. Here also it happens with the help of lysyl hydroxylase. I'm going to say about that later. Covalent modification. See, along with collagen, I'm trying to say some types of post-translational modification. What's the word? Post-translational modification. Proteolytic cleavage, aggregation, covalent modification, and one more. Some proteins are synthesized straight chains, and then they clump together or coil together to form a functionally active protein. And for that coiling, you need another type of compound called as chaperones. What is that? Just not the word chaperone. So another type of modification is folding. Okay, folding, protein folding. So these are the 4 major types of modifications happens after the protein synthesis. One is proteolytic cleavage, another one is aggregation, covalent modification, and folding. Here in the collagen maturation, we see 3: proteolytic cleavage, aggregation, and covalent modification. And another protein we see, some other protein we see this one folding with the help of a protein chaperones. Any defect in that can lead to a number of diseases. One of the diseases is actually our Alzheimer's disease. Is one of the reasons for Alzheimer's disease is the protein misfolding. Okay, we have a number of diseases in that. One of the examples I said, okay, is Alzheimer's disease. Fine. So along with the collagen, I'm trying to say some about its modification. There, along with that, about today I'm trying to say, I'm supposed to say about translation also. So just I use the word post-translational modifications. Fine. Now, 2 disorders related to collagen. See, here is the one, their hypermobility of the joints. You can see this, hypermobility of the joints. Skin, very loose. These are things what we see: abnormal tissue fragility, external joint mobility, hyper extensibility of skin. And this is because of our modification, our lysyl hydroxylase or lysyl oxidase. These deficiency, these are defective. So the type 3 collagen is defective. That's the reason behind the Ehlers-Danlos syndrome. One disorder related to collagen. One more disorder I will show you. The birth, at the time of birth, no, the bonds will not be formed properly, very brittle. We have osteogenesis imperfecta. It's called as osteogenesis imperfecta. There, at some sites, glycine will be replaced by cysteine. So, a defect of type 1 collagen. Osteogenesis imperfecta is defective type 1 collagen in the bond. So, the triple helical structure is disrupted. There will be more glycosylations in that. That leads to osteogenesis imperfecta. So, two disorders, Ehlers-Danlos syndrome and osteogenesis imperfecta related to the collagen, our major protein in our body. Now, I am coming to the mineral metabolism. First one about the calcium. What's the normal level of calcium? Normal level of calcium is 9 to 11 milligram per deciliter. You should study that. And you should study some normal values of phosphate also, 2.5 to 4.5. That value you should study. And then more than that, what are things you should know? First, the thing is, calcium, even though it exists in the 9 to 11, in the blood, it exists in 3 forms. I'll come to these terms. This one. Reference range is around 9 to 11. It exists in 3 forms: ionized form, protein-bound form, and just complex form. It's complex with carbonate, citrate, and phosphate. And this ionized form comes around 50%. That means 9 to 11 means 10, no value. In that 5. Around 5 milligrams will be in the ionized form. And that is the one is metabolically active. Now, this question. We know that when the calcium level reduces, that leads to tetany. You know it. Now, 2 or 3 portions are combined together here. One, you should know that tetany is because of hypocalcemia. Second, calcium level reduces leads to this one. So, normal calcium level 5. And that takes 3 forms: ionized form, protein-bound form, and complex form. And that ionized form is 5. That's the normal one. Now listen, whenever there is acidosis, we have protein. Let this be the protein. In acidosis condition, we'll be having a lot of positive charge. Extra. Because acidic, right? Express positive charge. So the calcium ion will not be able to bind it. So in acidosis, what happens? The calcium level in the blood will increase. Which calcium level? Ionized form will be more than the normal. Okay, total will be around 9 to 11 only. But in that, we'll be having around, not instead of 50%, it will be 60 or 70. Now, in the case of alkalosis, what happens? Alkalosis, protein will be having more negative charges. So, more calcium will bind to it. So, when more calcium comes and binds to it, you know what happens? The level of calcium in the blood reduces. Which calcium? Ionized calcium. So the patient will be showing some symptoms of tetany. Now I'll tell you, most of you will be in the dental colleges. You might be going to be my teachers. See, you might have heard this one. Some students, when they become nervous, suddenly they'll be having some carpopedal spasm. Why? When they become nervous, they'll be having high rate of respiration. So all the carbon dioxide goes out. They'll be having respiratory alkalosis. So nervousness, respiratory alkalosis, ionized calcium level reduces, leads to tetany. I hope you can correlate in that way. So just understand that about the calcium level. And now coming back to our topic, calcium level is maintained mainly by, we have some hormonal factors. What are the hormones? Our vitamin D itself acts like a hormone, calcitriol, PTH, and calcitonin. In that, calcitonin is hypocalcemic, PTH is hypercalcemic. This is hypocalcemic, this is hypercalcemic. Increase the calcium level. Fine. So this was about the calcium. Okay, now we have done factors of calcium level, hormonal, non-hormonal. And that we have non-hormonal factors are acidosis, alkalosis, serum phosphorus level, children, serum protein. Just now I said all things. These all together can reduce the, this one, can change the protein level, calcium level. Of course, the hormonal factors, as I said, this one, calcitriol, PTH, and calcitonin. Yeah. So these questions answer alkalosis favors binding of finest calcium to protein, decrease the level of finest calcium, so it leads to tetanus. Now, iron absorption. Second, going to the second mineral, one important mineral, iron. Absorption decrease in presence of. Okay. About iron, the most important thing about iron you should know is iron is a one-way metal. One-way mineral or one-way metal. Why you call for that? It can be absorbed normally. As per our need, it will be absorbed. But only through the bleeding process, only it will be excreted from the body. It will not get excreted from the body. So we call it as one-way mineral, one-way metal. And one more point, related to absorption. The iron level in the blood, in the body, is regulated. Iron content in the body is regulated at the level of absorption. Means what? Whenever we are in need of iron, iron will be absorbed. Otherwise, it will not. Simple example, simple way to understand this one. When you have iron tablets, next day's morning, feces stool will be very dark in color. Because only few will be absorbed, the remaining will be excreted out. The minimum we say RDA is around 10, out of which only 2 milligram will be absorbed. Okay, about absorption part. So this is about this one. And this one. If the extra is there, it will not be absorbed. So that we call this by a theory called as mucosal block theory. So iron absorption, understand that way. That's the level at which the iron content in the body is regulated. Iron is a one-way metal. It will be absorbed as per the need. It will not be filtered out. So that's why continuous, prolonged this one iron, our blood transfusion can lead to iron overload, can lead to hemochromatosis. Iron overload leads to a condition called as hemo. So our thalassemia patient, anemia patient, hemolytic anemia patients, hemochromatosis. In them, what we do, we'll be asking them to have the blood transfusion. No other go. And continuous blood transfusion, what happens? That iron will be stored there, that leads to iron overload. One of the type we call it as hemochromatosis. When we use a hemochromatosis, that's mainly about a disorder, a genetic disorder in the regulation of absorption. Okay. Now I'll say something about the regulation part. Here, anyway, as vitamin C is essential for absorption of iron. So any anemic patients, we give iron tablets. Along with that, vitamin C also should be supplied. We ask them to have that because vitamin C help in the absorption. I'll show now about iron absorption, but just see. Each is important. So that's why I'm trying to say about industrial lumen around 20 milligrams is our RDA. Iron, what all iron, see, when we keep the apple cut and keep, it will become brown in color. Black is brown, dark is brown. So because iron become ferric ions. This iron is in ferric state. Ferric. This cannot be absorbed. Then what happened? That iron should be converted to ferrous. That is done with the help of vitamin C. So that's the function of vitamin C. Vitamin C help in the conversion of ferric ions to ferrous. Then this ferrous ion through a transporter DMT1, please note, a transporter, this Fe2 plus get absorbed, stored there in the cells for oxidase, converted to Fe3 plus and stored as ferritin, one storage form of iron ferritin. Whenever needed, with the help of peroxidase and with the help of another transporter, we call it as ferroportin, it is transported to the blood. Again, for peroxidase, then ferric ions, part of your transport of iron as transferrin. So another word, ferritin, transferrin, then goes to bind to some receptors there, will be taken up, stored in the cells as ferritin. Now the point, how the process is controlled. From liver, a protein is inside, peptide is produced, hepcidin. So this is juvenile side, this is okay because of cells, basolateral side, fine, leave that. In the liver, a peptide is produced, hepcidin. When the iron content is more, this hepcidin will be produced. That will reduce the level of DMT1 and ferroportin. So these all are important for you. These terms, iron absorption, there comes a word DMT1, divalent metal transporter 1, ferroportin. So using this, it is absorbed. Its level is controlled by a protein called as hepcidin from liver. That's a major way of regulating iron absorption, our mucosal block theory. My question here was about vitamin C that help in the conversion of ferric ions to ferrous, the ferric reductase enzyme. But I said other question that can come, mucosal block theory, DMT1, ferroportin, hepcidin, and peroxidase is a copper containing this one. So without copper, iron metabolism will not take place. So copper deficiency leads to iron deficiency. That's what I want to say. Peroxidase is copper containing enzyme. Fine. Now another question. A patient suffers. So iron, just I'm leaving the iron part. I said about hemochromatosis also. Hope you understood that part. Hemochromatosis. And then, okay, iron, I said about ferritin, I said about transferrin. And one more term, transferrin. There, I want to say one more word. Iron binding capacity, called as total iron binding capacity in the blood. Whenever the iron deficiency reduces, the capacity for the iron binding, that increases. So one parameter that increases during iron deficiency is total iron binding capacity, TIBC. So we do iron, the level of iron, we try to find out the level of ferritin, we try to find the level of TIBC. These three are biochemical parameter we use to analyze the iron level in the blood. Iron itself, ferritin and TIBC. And TIBC will be increasing deficiency. These two will be decreasing during deficiency. Understand? Okay. Let's go into the word, the another mineral, zinc. Zinc, okay. Patients suffered from hypogonadism, failure to thrive, loss of taste, unable to maintain stability. These show the deficiency of, I already said the answer, zinc is the answer. Please note that during the COVID time, we're all made an awareness that the patient should take zinc plus vitamin. What all are the functions of zinc? Zinc has a lot of functions. We'll say one by one now. I'll show you one by one. You might be knowing this word. Whenever a patient is having diarrhea, severe diarrhea, we give zinc sulfate. They'll be able to maintain the mucous membrane. They'll be reduced diarrhea because electrolyte absorption, mucous membrane integrity. That thing you might be knowing. You might be remembering. What are other things? Let us see. Zinc containing protein in the saliva. Gastric. So taste. Spermatogenesis. Zinc is essential. I'm sorry, the spelling mistake. Secretion of insulin from the beta cells. You need zinc. Carbonic anhydrase that help in the dissociation of our carbonic acid. Real regulation of pH. That contains zinc. ALP, alkaline phosphatase. For the bone and teeth formation, we need zinc. This one, it contains zinc. Then intestinal fluid transport, as I said that, regulate the absorption of fluids and electrolytes from the intestine. So counteracting the fluid loss associated with diarrhea. Mucosal integrity, regeneration, maintenance of intestinal lining during the diarrhea time. Next one, this was the one important reason why we were asking the patients to have zinc of it. See this word, zinc for our monocytes, for cytokine productions. Very crucial for the T lymphocytes. So the immune system to function, we need zinc. And it's an antioxidant. See, some enzyme containing that. Antioxidant, zinc containing enzyme. Or catalase, peroxidase. So clearing of the pathogens from the body. So these all are functions. Then how can a patient survive, thrive without zinc? Spermatogenesis, hypogonadism. So all other things. Now, hope we can understand now the things. Unable to maintain stability, failure to thrive. These are hypogonadism. These all are deficiency of zinc. So these are the functions of the zinc. A lot of functions are there for the zinc. Even though it's a micronutrient. We know that, no? If the need is more than 100 milligram, we call just macro. Less than that, we call just micro. So zinc is a micro element. And here about the chromium. Chromium is important for the insulin sensitivity. Insulin secretion, insulin sensitivity, chromium is important. Copper, I said about peroxidase. Anything else about copper? Of course. Copper, related to copper, we have two disorders. One is Wilson disease. Wilson disease is all about copper accumulation. Okay, copper increase copper. Increase in copper. And we have another disorder, MENS or Menkes. That's the deficiency of copper. Two disorders. Related to this one. Copper is essential for peroxidase or for iron absorption, so anemia can occur in that way. Copper is important for lysyl oxidase. Very important part. Our collagen and elastin, lysyl oxidase. So your blood vessel wall become thinner and the blood vessel wall may have a ballooning and thereby break down. We call it as aneurysm. You might know this word. Aneurysm. Sorry, see there. Aneurysm. That can happen in Meng disease. Why? Because that collagen interlink will not take place, so it will be fragile. So there will be a bulging, ballooning of some portions of the patient's blood vessel. We call it as aneurysm. So Meng disease, anemia, we can say. Aneurysm is another one. And always say Menkes, kingy hair. Kingy hair is there. That means breakable hair. So brittle hair. Menkes, kingy disease. We call it like that. We say like that. Okay. So these are some symptoms related to copper. There are a number of enzymes containing copper. Okay. Our heme synthesis, our ALS synthesis contain copper. So heme synthesis we need. And peroxidase contain, I said that. Lysyl oxidase. And our cytochrome oxidase. Hope you remember the word electron transport chain. We have a complex 4. Complex 4 contain 2 molecules of copper. So copper deficiency can lead to the energy production, then decrease in energy production through electron transport chain. That's another function of copper. So that's all about the copper. It's about zinc also. So zinc I said. Now going to the nucleic acid metabolism. There first I say about DNA. DNA contains 2 types of bases, purine, pyrimidine. Purine are adenine and guanine. And purine, adenine always have mixed 2 hydrogen bonds with another pyrimidine, thymine. And guanine mixed 3 hydrogen bonds with cysteine. So ATGC. So the 2 pyrimidines are thymine and cytosine. Purines are adenine and guanine. Now the answer is here. Here we have uracil. Uracil is present in the RNA. You know that. You know it. That's the simplest question. DNA structure, as you know, double-stranded helical structure. Double-stranded helical structure. Watson and Crick model. You know the word. Watson and that's the simplest one anyway. +2 knowledge only. Watson and Crick model. And it contains number of purine is equal to number of purine, pyrimidine. Pyrimidine, you know that. And the rule we call it as Chargaff's rule. You know it. Chargaff's rule. That also you know it. Okay, then two chains are there. They are anti-parallel. One is from 5' to 3'. Other will be from 3' to 5'. So we call it as anti-parallel. And they are linked by hydrogen bonds. They are complementary. Both strands are complementary, not they are equal. Complementary. I hope you understood that. That means what? If a chain contains adenine, thymine, cytosine, guanine, the next chain will not contain adenine here. It will be thymine. Here it will be adenine. Here it will be guanine. You know about the base pairing rule. Guanine, cytosine. So they are the chains that are complementary and the chains are linked by hydrogen bonds. You know it. Okay, between the chains are hydrogen bonds. So these are some ideas about DNA structure. And please note we have 3 types of DNA structures. The same helical structure. The common one is B form. We have A form and Z form. Please note we have mainly there are 3 forms are there of DNA structure. B form, A form and Z form. Okay, please note that. Okay, that's all about the DNA structure in general, basic ideas. Now, from RNA, by the process of transcription, we produce RNA from the DNA, and they are synthesized by the process transcription. And we have mainly 3 types of RNA. We know that major ones: mRNA, rRNA, and tRNA. In that, rRNA is produced in the nucleolus. That's one difference is there, nucleolus. Other one, the nucleus itself. Okay, and more than this, we have some more RNAs. Please note, we have for the transcription purpose. During transcription, mRNA synthesis, first straight away mRNA is not produced. Produced is hnRNA, heterogeneous nuclear RNA. That's the one converts to mRNA. So hnRNA is a precursor of mRNA. That's one thing. Then RNA, during its modification, you need an RNA called as the normal mRNA, hnRNA conversion to mRNA. There's some modifications. At that time, you need an RNA called as snRNA, small nuclear RNA. hnRNA to mRNA conversion, I'll be coming from that word. There is one process called as splicing, removing some part. That for the splicing process, we need another type of RNA called as I said earlier, snRNA. So then another one from mRNA to protein synthesis. Some processes are related by another RNA called as microRNA. So these are some different types of RNAs, okay? And tRNA, what is the tRNA? That will be carrying amino acids to the protein synthesis site. At the same way that identifies a codon. I'll come to that word codon. mRNA contains this information for protein synthesis. rRNA is a part of ribosomes. Okay. Now, this adenine guanine on its breakdown process, what is the end product? End product is uric acid. Sorry, I didn't write that. I didn't mention it. Uric acid is the end product of purine degradation. So I'll just again say protein degradation. What's the end product? Protein metabolism. Protein metabolism, we get amino acid catabolism, we get ammonia. Ammonia then converts to, just now we said, urea. So the urea is the end product of, we say, either we can say amino acid catabolism or simpler words, protein catabolism. Purine catabolism, the end product is uric acid. And creatinine we get from creatine. Creatine. By removal of one water molecule, we get creatinine. So difference is one water molecule. Fine. Here, uric acid is the end product of purine degradation. There anything about uric acid to be known? Yes. Uric acid thing is uric acid accumulation leads to a condition called as gout. Primarily, first we see the first joint. We have a gouty, we call it as gouty arthritis. The word arthritis itself meaning arthro means joint, inflammation of joint. The first joint to get affected is our first metatarsophalangeal joint or first big toe. First big toe is the one will get affected in gout. We should know that gouty arthritis. We have primary gout and secondary gout. Primary and secondary gout. Primary is some abnormality of enzymes. Abnormality of enzymes or some defect in enzyme, deficiency or abnormality. But secondary means because of some other diseases. Can be cancer, renal diseases like that. Tissue breakdown is increased. Then we have gout. So just know the word about gout and the primary and secondary gout. Okay. Going for next. What's the process involved in the conversion of DNA to RNA? It's nothing but transcription. Transcription. Other things are related to the transfer of back DNA from one to another. Other one. So transcription is the answer for this one. And another thing we should know is translation for protein synthesis. You know it. This is what I was trying to say. In transcription process, using an enzyme RNA polymerase, we help in the conversion of one DNA template strand will be converted to RNA. RNA is single stranded, DNA double stranded, DNA first denature using the helicase enzyme, then convert to RNA. Then RNA is produced. For that, we need the enzyme RNA polymerase. This RNA polymerase also contains zinc. Okay, zinc is another function of zinc. It helps. It's a component of RNA polymerase. Okay, after the RNA synthesis, as I said, when mRNA is synthesized, first produces hnRNA. Just now I said hnRNA. Okay, this hnRNA will be having some region but are required for, I'm just putting it this way, required for protein synthesis. We call it as exon. We'll be having different exons. These are non-coding part. We call it as intron. Introns are non-coding part. Exons are coding part. So what happened? During modification, all this will combine together. Our exon 1, exon 2, exon 3 combine together and form the mature mRNA. In this process, what all happens? At the 5' end, we attach a cap. 7-methyl guanosine. Usually between the nucleotides we have a phosphodiester bond. But here alone, triphosphate bond. So 7-methyl guanosine attached at the 5' end using a triphosphate bond. In the tail, in the 3' end, you have a number of AAA. That's attachment. Then other modification. Then the removal of introns and addition of joining of exon, that we call it as splicing. So please note, splicing means it's not slicing, it's not cutting. Splicing, the word means joining. Joining of exons. This is exon 1, exon 2, exon 3. Joining of exon after the removal of intron, we call it as splicing. For this splicing, as I said earlier, we need an RNA called as snRNA. Hope you remember that. Okay. So the process of transcription modification after transcription, we call it as post-transcription modification, mainly for snRNA. We have 5' capping, 3' poly-A tailing, splicing. Glycosylation is the one that happens after protein synthesis. That is post-translational. So this is post-translational. All other three are post-transcriptional modifications. So these are about modifications of that, about the how to convert hnRNA to mRNA. So please note these terms: hnRNA, mRNA, splicing, snRNA, 5' cap. What is 5' cap? How they are linked through triphosphate. So we say 7-methyl guanosine triphosphate is attached. We say like that. That's 5' cap. A number of adenine at the 3' end. These are some modifications. Okay. That's about the RNA modifications. Over. Now comes the word genetic code. In mRNA, you have information to synthesize protein. About each amino acid, there are some information. For example, AUG, that 3 nucleotide sequence. We call it as, that codes for an amino acid, methionine. Then U, that stands for phenylalanine, like that. So 3 tri-nucleotide sequence in mRNA. That gives information about one amino acid is called as genetic code. That's the first point. Now the question is, all are true of genetic code except whether it is generate, universal, punctuation, non-overlapping. What do you mean by these terms? Anyway, the answer is punctuation. I'll come to that now. I'll just show you. Let this be your mRNA. Think that this is an mRNA. Any mRNA, they start with this AUG. That's the initiation codon, AUG. First code for methionine. So first we'll code that code, we'll decode it. Second, after that, we should decode the second 3. The first 3 we decode. Then we should decode the second 3. But we can't do that. That's why I put it in red color. We can't do this. There's non-overlapping. We can't overlap. AUG, we first read the AUG, then that is over. Then you should read next 3. 4, 5, 6, CAA. Okay, so that's the next one. So that means there's no space, no punctuation between. So non-punctuating. So genetic code is non-punctuating. This AUG is there in everything, every body, every animal, every organism. AUG means methionine. So the word universal. And there is no, as I said, non-overlapping. So the word is consecuting. Okay, consecuting. Next word. After AUG, you can't, okay, we see, we just, next 3 is over. We won't, I don't want to read next 3. I'll go for these 3. No. It should be continuous. It should be continuous. So then this is what happens. See, it should be next one. So this is another feature. Non-punctuating, non-overlapping. Non-overlapping means what? Non-overlapping in the sense, no gap in between, consecuting. After 3, next 3. That's what it by non-overlap, consecutive. Then no punctuation means no gap in between. No gap. It should be continuous. Non-continuous. Okay? It should be continuous. I'm sorry. It should be continuous. Non-punctuating. Then it should be universal. Now go back to this one. And then one more. U. That calls for, okay, now this in this way it comes. CCA, proline, CCA, proline, ALA. This one, GG, glycine, UUU, phenylalanine. Here, sorry, UUU. That's a phenylalanine. UUC also phenylalanine. So that means, see, if there is UUU, it codes only for one amino acid, phenylalanine. So we use the word another property of codon. We use that unambiguous. No other meaning. Unambiguous. Only one meaning. Unambiguous. Okay. But at the same time, you code for phenylalanine. No doubt, no other can be coded. But at the same time, UUC also codes for phenylalanine. So the value of this is degenerated. So another feature, degeneracy. Degeneracy of codon. That's another feature of codon. So these are the features of codon. Degenerate. That means one amino acid has more than one codon. Universal. We said no, it should be non-punctuating, non-overlapping. So here the answer is punctuating. That's what the answer is. Okay. Now I'll show the features of it. And there are totally how many codons? 64 codons are there. One AUG is the initiation codon. 3, this one, our termination codons, UAA, UAG, UGA are the termination codon. Unambiguous, I said, undoubtful, specific. That means if one codon stands for only for one amino acid. See, there are totally 64. 3 are termination, then other one 61. The remaining 61. There are 61 codons, but 20 amino acids. That means what? Some amino acids has more than one. Our leucine, serine, arginine has more than, they have 6 codons. Each has 6 codons. So that is called as degenerate. So these are the features of genetic code. Then comes a theory. There's a question in your question, the earlier previous question paper. Hypothesis, wobble. Wobbling means what? Shaking. It is unsteady. See, here, codons are present in the mRNA. U. These codons are identified by the anticodons in tRNA. tRNA we have anticodon arm. They'll be having, you always start with, okay, A. So they are by base pairing rule. But understand that UUC also calls for the same. The same tRNA will be able to understand this fellow. You know why? Because there comes a rule. There comes a wobble hypothesis rule. See, whenever we number it, the mRNA, we number from here. This is 5' and this is 3'. So this is the first nucleotide of the codon, second nucleotide of the codon, third nucleotide of the codon. And anticodon, this is anti-parallel. So here from 5' and so this is the first nucleotide of anticodon, second nucleotide of anticodon, third nucleotide. Now comes the theory comes. The pairing between the first nucleotide of anticodon and the third nucleotide of the codon, it always does not obey base pairing rule. So what happened? The same anticodon can pair with different codon. This is called as wobble hypothesis. That's the reason for degeneracy of codon. Anticodon pair with the codons. Here you look. This is for arginine. All the 3 are for arginine. CGA, CGU, CGC. All 3 are for arginine. And you have, this is the anticodon. GCI. I stands for inosine. Okay. So this GCI will be able to code with these all 3. The first codon, first nucleotide of the anticodon is inosine. That can code with C, that can code with U, that can code with A. So the pairing, so this is wobble hypothesis. The pairing between first nucleotide of anticodon and third nucleotide of the codon does not always obey base pairing rule. Okay, that is wobble hypothesis, please not. So the same anticodon will be able to pair with different codon. Because of which, several amino acid has more than one codon. Other word, degeneracy of codon. So degeneracy of codon is explained by wobble hypothesis. That's what you should know. Degeneracy or degenerate, that property is explained by wobble hypothesis. And in simpler word, this is what? Same anticodon can code with different codon. Hope you understood. That's about the codon part. Second, mutation. Any change in the nucleus sequence is called as mutation. See what happened? A, U, A, G, UAA, UAG, UGA. These three are termination codons. Okay. Then when this codon comes, protein synthesis stops. Okay. Now the point. In some case, in an mRNA, this was a codon UAC, a codon. They are coded for amino acid. Because of some changes, this UAC converts to, so this was giving you some sense for an amino acid. Okay. This was able to give an amino code for an amino acid. Now, because of mutation, this become UAA, C become A. So what happened? This will not give any sense. Here this one, our protein synthesis terminates. So the word nonsense mutation. So when an amino acid codon is converted to a stop codon, that's called as nonsense mutation. We have different types of mutations. You know the word transition, transversion and all. Transition. Whatever transition means, when adenine, a purine, mutated by another purine. Or when a pyrimidine mutated by another pyrimidine. That is called a transition. When a purine is replaced by a pyrimidine, we call it as transversion. Understood that. Then what is silent mutation? See, U stands for phenylalanine. Because of some mutation, it becomes UUC. There is mutation, but there's no change in amino acid. Both codes for same amino acid, phenylalanine. That's called as silent mutation. Hope you understood that. That's about the some mutation part. And what is frameshift mutation? Just in a codon, these EHR nucleotides, think that this is one codon, this is another codon. One nucleotide got deleted. Then what will happen? These three will be first codon. Then another three will be another codon. So totally change will be there in the protein. So that's called as frameshift mutation. Frameshift mutation occurs because of deletion or insertion of one nucleotide. So whole codon will change. That's about the mutation part. Okay. Now come the last question for today's topic. Restriction endonuclease. What is it all about? Our body, in the body, to break the DNA, we have some scissors. We call that molecular scissors. Molecular scissors. Scissor. I hope you know the word scissor. Our restriction endonuclease enzymes are molecular scissors that can cut the double-stranded DNA. Okay. That is essential to make, see what happened? One portion of the DNA, we cut that and then we attach to some other DNA. We add over there. So this is one DNA. This is another DNA. We are adding that. So two DNA from different sources combined together. So they are combined together from a new one. We call it as recombinant DNA. For synthesis of recombinant DNA, the major enzyme required is restriction endonuclease. But not only that, not only restriction endonuclease enzyme, we want to link together. So we have a molecular glue. This is, if there's a molecular scissor, we have a glue also. molecular glue, that is nothing but ligase enzyme. Ligase enzyme. And for to synthesize, to manufacture insulin, we want to get the RNA, we get the mRNA of insulin. From there, we will make the CD, this one, rDNA. For that, we need an mRNA. From there, we synthesize DNA. So just opposite to the transcription. So the enzyme reverse is reverse of transcription, reverse transcriptase, reverse transcriptase. So these are the three major enzymes that we require for rDNA technology. Hope you understood that. Just here the question was about just the enzyme, restriction enzyme that cut DNA, double-stranded DNA. It's also called as molecular scissor. At the same time, we need an enzyme that can link together. We call it as molecular glue. Like scissors, we have glue, ligase enzyme. And in some recombinant technology, we take the, instead of taking DNA, we take the mRNA. From there, we make small complementary DNA. For that purpose, we need another enzyme, reverse transcriptase. So here ends our today's topic for the molecular one. Some important questions of each topic has been taken. I hope you enjoyed and you understood the matter. Thank you.